Good afternoon. We're happy to see so many of you here once more for our next installment, Anti-Aging number 2. And we will continue our talk on telomeres, and we'll get a lot more detail. In the overview, we just sort of mentioned them. Now we're going to talk more about what they are, how they influence aging. We're going to introduce something called telomerase, and we'll talk about how stress is probably the #1 factor in all of this. So first of all, the whole idea of why telomeres matter is that cells divide. And on the first slide here, we have a picture of mitosis. And that simply means that that's when a cell makes a complete copy of itself. So the first part of that is to replicate the DNA inside, and then the DNA, when you have 2 copies, each copy finds a separate half of the cell, and then the cell splits, and you have 2 separate cells with a complete copy of the original, called diploid cells, meaning they have a complete setup of that DNA. There's a different kind of copying called where you end up with haploid cells, and that's where you don't make a complete DNA replication. You just split it down the middle, and then you separate the cells with half, and that's what makes up an egg cell or a sperm cell. And we'll talk a little bit more about those. They become interesting because these haploid with half the genetic, they behave differently than a diploid. So when we have this cell division take place, there is a peculiarity that we mentioned, that the DNA cannot copy 100% of all of the different base pairs. It is not possible due to the mechanism, and they knew this very early on. The Watson and Crick, who discovered the structure of DNA, they also figured out how DNA replicates, and they said this doesn't make sense, because with this mechanism it's not possible for the DNA to copy 100%. It's going to lose a few base pairs at the end. And if that was the case, then how can you copy all the information over to the next cell if you lose a little bit of the chromosome? And telomeres is the solution, it's nature's solution to this. Because telomeres is the last portion, the last part of the chromosome is called a telomere. And the telomere is DNA material that doesn't code for anything. It's a repeating nonsense sequence. So you can lop off a few base pairs each time, and you're not losing anything that would code for anything. You're not losing anything that would code for proteins. And one of the best metaphors or pictures for this is a shoelace. And there is an actual term for that little protective cap. It's called an aglet. So now you learned something useful today. And it's very much the same thing, that the aglet keeps the shoelace from fraying and getting damaged, and that's exactly what the telomere does. It keeps the end of the chromosome from fraying and getting damaged. So these telomeres are kind of funny, and this is a very, very young, very new science, and there's a lot of theories, and they're finding things every day, and they still have a very incomplete picture of how this works, but it's getting more and more interesting. So Elizabeth Blackburn she did some research on this in the 1970s, 1980s, and she got the Nobel Prize in 2009 when they realized how profound this research was in relevance to aging. And on the rough model, what they found is the sperm cell and the egg have about 15,000 base pairs of telomeres. The length of the protective portion is 15,000 base pairs. And then the cells divide to make a fetus, and by the time you're born, you have about 10,000 base pairs left. And you can't get down to 0 base pairs because there needs to be a certain length. We'll talk about some of those reasons, but you have to have a certain length of that telomere for the chromosome and the cell to work, for the cell and the chromosome to know what's the end, what's up and down on the chromosome. So by the time we get to about 5,000 base pairs, then the cells stop being functional. They don't know how to replicate the DNA anymore, and they just stop. They call that state senescence, which basically means that the cells won't divide anymore, they won't do much of anything. And when a large portion of a person's cells reaches senescence, then we're going to die very shortly thereafter. And some of the things that happen is that chromosomes can break. You can, for various different reasons, they can get damaged and they can break. And the cell has the capacity to glue them back together. And this is really, really important, and this is illustrated on the next page with a little blue and red there, that if it breaks, the telomere will tell the cell what is the end of the chromosome. So you're not going to try to glue it back together at that end. But anything that doesn't have a telomere is fair game for gluing. So if the telomere gets too short, then it might get glued back together in the wrong place. And that would be the picture on the right. And now you have a completely dysfunctional chromosome. That one is not capable of replicating even once. If the body tries to duplicate that one, it's just going to rip apart and shatter, and there won't be anything but chaos left over. So there are many factors involved in the function of these telomeres, and that's one important aspect. So what Elizabeth Blackburn, she calls the chromosomes get sticky if they don't have the proper length of the telomere. This creates instability in the chromosome. The length of the telomere provides stability, and instability is one of the hallmarks of cancer. They have a lot of research, a lot of evidence now that when the telomeres get too short and the genome gets unstable, that's actually one of the things that lead the cell to mutate and become cancerous. What they're finding that's really interesting is that aging seems to be programmed. It seems to follow a pattern. And we can tell that because most of us age at approximately the same rate. But when some mechanism in the body fails, then aging can occur much, 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 much faster. And in progeria, in a normal cell, there's a gene called lamin which codes for a protein, and don't have to worry about the details, lamin A, which maintains the structure of the cell nucleus. And as the telomeres shorten, the cell starts making a toxic version of this protein called progerin, which accelerates aging. So it's not just whether we have a telomere or not, but how long it is determines sort of how well the cell works, how well the structure of the cell nucleus is. And the shorter the telomere, the more progerin the cell makes. Well, in this disease, they have a mutation that they're not making any of the lamin A. So all of their proteins look like the progerin, like the aging protein. So these kids age about 5 to 7 times faster. And that's why they start balding at 5 years old. They start getting arthritis and heart disease. And it's not just the looks, but every organ of the body, every tissue that they can measure, every disease that people get in their 70s, these kids get in their early teens. And they die as teens from old age. And everything that they've studied about chromosomes fit exactly with that aging process, because they have short telomeres to start with, and their telomeres wear shorter at a very, very fast rate, and they don't have the enzymes to protect the telomeres. And so it seems like the telomeres are sort of flexible. And this brings us to something very interesting, that after Elizabeth Blackburn had found out about the telomeres and the structure of the telomeres, they found that it didn't seem completely consistent. It seemed that in some cases it appeared that the telomere could regrow. And then they figured there has to be a mechanism, there has to be a gene, there has to be an enzyme, there must be something to make it regrow. And she started looking for this. And I think she had to look for about 8 years. And that's part of why she got the Nobel Prize. But eventually she found a gene that codes for an enzyme called telomerase. And this is an enzyme that will actually lengthen the telomere and make the cell more youthful. And that was quite astounding, because all of a sudden, aging was not something that just moved in one direction. It really was possible to make it reverse and go the other way. And one way that they figured this out was by studying, like I said, the sperm cell, egg cells and sperm cells. These have cell division, but they make a haploid cell. And when the DNA is in its half configuration, called haploid, like in a sperm cell, now the telomerase gene is active, and it protects that telomere indefinitely. That sperm cell is 100% immortal. It can divide any number of times and not lose an iota of that genetic material. So the mechanism, this becomes really interesting, because now aging isn't inevitable. The mechanism to regrow telomeres and keep cells youthful forever is there. But as soon as the cells combine, as the sperm and egg combine to make a whole cell, then now this telomerase gene becomes partially inactive. Not completely inactive, because it seems to be able to increase or decrease activity. So it can speed up or slow down aging. But there is an enzyme that does this. So some of the research that they have done is they have taken individual human cells, like skin cells, that are old skin cells, and they have injected them with telomerase, and they have reversed the age of the skin cells. Not just made them a little bit younger, but if they keep adding telomerase, they can revert that aged skin cell all the way back to a stem cell before it even differentiated to its origin cell. It's like a fetal stage cell. And another thing that they did with mice is they engineered some mice. They fixed their DNA so that the telomerase gene was completely blocked. And now these mice started aging very, very fast. Within a couple of months, they let them reach adulthood without this enzyme, and they were aging very, very quickly. And then they figured out, because they had done this mutation on purpose. They had fixed it so that they could give the mice a chemical and reverse the change, the damage. And so they had these mice, they were born, they were allowed to reach adulthood, and at that point, these were very, very old, decrepit mice. They were way past the point of fertility. They started getting age-related brain shrinking. Their brains were down to 75% of an adult mouse. They had arthritis and organ failures, and their neural development started shutting down. Then they gave them this chemical to activate the telomerase again, and these mice became younger. And their brains regrew, they started developing new brain cells, they reversed organ damage, and they became fertile again. Okay? So the mechanisms that we're talking about are that you're not just talking about sort of cosmetically or partially fixing different aspects. You're talking about turning back the entire clock. So it's pretty exciting stuff. And even if we're not at the point where we can turn back the entire clock, we have figured out that this telomerase is not a black or white thing. It's not entirely on or off all the time, and there are certain lifestyle things that we can use to affect it. So they took a group of people, and the research, I didn't read the actual article, I just heard it referenced by Dr. Blackburn, and they took a group of people who were caregivers, and they had them rate how stressed they were in their role of caregiving. And then they divided them in two groups, the top stressed and the most stressed half and the least stressed half. And they measured their telomerase activity and the resulting telomere length. And they found a very statistical, clear as day and night, that the people that felt more stressed had less telomerase activity and shorter telomeres. And the difference was when compensated for actual age, the difference was as much as 10 years of biological age, just from how their self-reported level of stress. And it became even more significant when they included how long, for what period of time they had been under this stress. Then they took, on the same group of people, they measured cortisol and adrenaline and noradrenaline. And these are basically, all you have to know is these are stress hormones. They are the hormones that the body makes in response to stress, and they shorten the telomeres. And again, when they took the top half of people and the most stressed people and the least stressed people, the top half had the more stressed people had much higher levels of cortisol and adrenaline and noradrenaline. And of course the resulting shorter telomeres. This testing that she quoted is very close to the testing that we do in here. So the stress test that we do and the stress test that they did were quite similar, and it shows a dramatic difference in the telomerase activity. So what they had people do in this, and there's a slide for this one, they measured a resting heart rate level, resting heart rate variability level, which is something we measure as well, and they measured a pulse pressure level. So, and then they compared between the highest 25% and the lowest 25% of And this was where they had measured the telomerase activity in the people. So the people with the 25% with the highest telomerase and the 25% with the lowest telomerase, they compared the two groups. And then they had them in a resting baseline reading, and then they told them, you're going to have to make a 5-minute speech into a video camera. And then they left them alone for 5 minutes. And this was the anticipation stage. And they saw how stressed were people just thinking about this event. And then they had them give the speech, and then they had them recover, and then they had them do a math test, a head counting math test, which is very similar to the type of stress test that we do. It's not exact, but it's the same idea. So if you look at the green and the red lines here, the people with the higher telomerase activity, the higher DNA repairing activity, their resting heart rate started at 70. And the people with the low telomerase activity, the people with poor repair, started at 82. And then, when they told them they were going to make a speech, both of them jumped. One jumped 12 points and the other jumped about 10 points. And then they gave the speech and they jumped a little bit more, and then they had them rest, and the math wasn't quite as bad as giving the speech. But overall, you can see that the average heart rate was about 20 beats higher almost, 15 to 20 beats higher in the people that had better, or the heart rate was higher in the people that had worse DNA repair. So the things that we measure in this test, the heart rate and the heart rate variability, it's not just, it's like you have higher heart rate, it has a direct effect on how well your body repairs at the cellular level and how long you're going to live as a result. And then they did the same thing, and they measured the heart rate variability. And here, it was the higher heart rate variability is good, because that means your nervous system is more flexible. It's responding better to the challenge. And then at the last one they measured was pulse pressure, and the people with the low telomerase, they had a huge spike in pulse pressure, means their heart went crazy in anticipation of giving the speech. So I think this is absolutely fascinating, and it shows how huge, how significant, how direct an impact it is that stress is on aging and on DNA repair and telomerase. So what can we do? This is the same as we had last time, but it's just want to round off and repeat this. What can we do as individuals? Well, we want to be willing to learn. If we don't know why, we're never going to stick with it. There's so many things out there we know we should do, and we don't do any of them because we should. As soon as we should do something, then we just put up our defenses, and rarely do we do it. But if we know why, and we know that we want to do it, now we'll have the excitement and the incentive to do something. That's the only thing that people will ever stick with. And why do we want to get adjusted? Because chiropractic is one of the most powerful ways of interrupting the stress response that has ever been found. And all the things that we talk about in this talk, all that stress can be reduced by the adjustment. It's not the only thing you want to do though, because we are whole ecosystems, and everything that the ecosystem needs, you have to provide in order for that ecosystem to thrive. So there's no quick fix. The chiropractic adjustment is not a quick fix no more than a pill is. But it's a very, very important component. Lifestyle, learn how to eat better, move better, feel better. On other talks I talked about what the requirements for a plant are. So we'll run that one again. Have you heard it? Yeah? What does a plant need? You have a plant, and if it's not looking too happy, what do you give it? Water. And then you give it some water, and it's still wilting, still not so happy. What do you give it next? Try some light. Exactly, take it out of the closet. And then you give it those two, and it's still not thriving. What do you think it is? Nutrients. Quality of the soil. So a plant has to have water, sunlight, and nutrients in the soil. So if you have 1,000 plants and you do an experiment on all those 1,000 plants, you divide them into different groups and you give every conceivable combination, you give 2 out of 3. Some of them you give them water and sunlight, no nutrients. Some of them you give them nutrients and so forth. At the end of a year, how many healthy plants are you going to have? 0. Because if something is required, you can't live without it. You can get by for a while, but you can never thrive in the long term. So humans are incredibly resilient, and we have nutritional and physical and emotional requirements. And we can focus on nutrition and skip the others. We can focus on exercise, skip nutrition. We can eat well and work out and feel horrible. We will never thrive with 2 out of 3. So that's why we want to talk about the whole ecosystem and what the ecosystem requires. And that's why we talk about eat better, move better, feel better. And feeling well is not something that happens by accident. It's a skill that you practice and learn. It's learnable, it's a practice. Then you want to reduce your toxic burden, and you want to do a cleanse a few times a year. Supplements are proving increasingly important, because in this world we're not really getting all the nutrients that we need, and the more stress we have and the more toxins we have in the environment, the more nutrients we need to compensate for it. The liver needs fuel to help detoxify. It needs the energy and the substrates to help clean us up. And exercise is not optional. We can never ever function optimally. We can get by for decades, but we cannot function optimally without all the components that we need. And exercise is very, very important. We're going to have a talk where we talk about exercise does 2 things. It stimulates your body and makes it and stimulates growth hormone, which is fantastic. And on the other hand, exercise is stressful and breaks you down. So the correct exercise is about finding the balance that gives you the best boost of growth hormone with the least amount of stress. So that's the trick to exercise. So that will be all for today, and I thank you all so much for coming, and any questions you might have, we'll try to entertain those. Thank you.